So, welcome to our first podcast. Hi, it's Ms. Lee. Today we're going to be talking about science in terms of methods and madness, focusing on things like experimental design and things like sperm and tails. So, by the end of this lecture, it's always good to kind of know where you're going. You should know what sperm stands for and why it's important. And I'm not talking about that guy. And tails, not the thing on the back of your dog's bottom, but why they relate to science and the methods and madness. So, right off, let's start off with sperm. What does sperm mean? Sperm means to state the problem or question, predict. Another fancy word for prediction might be a hypothesis. E stands for experiment. In this case, it's going to be our experimental methods. It's also going to include things like our procedures and materials. The R stands for our results, our data. What do you hope to get out of this project? And lastly, the M in sperm stands for make conclusions, which we'll go to in a little bit more depth later on. But here's sperm and what it stands for. It's basically an acronym, kind of help you remember the steps. Remember, science is not linear, but this is kind of an outline of what scientists would do. Alright, so let's start with the S. Again, we said the S stood for what? Yeah, it stands for stating the problem or question. So when you have a problem or question, we want to make sure when you're designing it that it's actually a SMART question. So what does SMART mean? It means that it's specific, meaning that it has to do with a certain organism or certain situation. It's not this vague thing like what is an animal. It's very specific to a cause. It's measurable, meaning you can go out and you can either use a ruler or other scientific tool. You could look at it with and record it and put it maybe on a screen and bring it back. It's measurable. Attainable, meaning you can get there. It's not at the bottom of the ocean where no one can go. It's relevant, meaning that we care about it. It's something that's kind of important. And lastly, it's timely. So unless you're doing a PhD and you have like 8 years to do this, you may want to make sure, hey, I can kind of do this in the time that I have. So if you have an hour to do this lab, you want to make sure that your question is something you'd solve in an hour. And isn't something that someone spends their whole life doing. If it is, and it's something you spend your whole life doing, then you might want to think about doing it for your PhD project. But in terms of a problem or question, we want to make sure that it's these things. It's specific, meaning it's relevant to the task at hand. It's tight. It's measurable, meaning you can go out and actually get some data. It's attainable, meaning you could do it. Relevant, meaning it has to do with a concept. And timely, meaning that you could do it in a reasonable amount of time and that it won't take you years and days, but it could actually take you a class period. So what does the P stand for? We said that P stood for predict. In terms of a hypothesis, we use an if, then, because format, and that actually goes with the format SIR. If is a claim. You're making a statement about what would happen. Then would be your evidence. So if it rains, then it'll be wet because, lastly, we'll end up with our reason. Rain is precipitation, which is water. So our prediction should be an if, then, because statement, because that matches up with SIR, a claim, evidence, and reason. You'll hear more about this in our class as we do more experiments, as well as in your English class, where they're looking at things like argument and logic. We'll also touch on this when we talk about controversial topics, such as stem cell research and things like abortion that may be socially charged, where we're looking for evidence, as well as things like evolution. Now we get to experimental design. We have just 3 kind of vocabulary words to keep in mind when you're designing an experiment. An independent variable is something that the experimenter changes, or the researcher changes. The dependent variable is the kind of thing that the researcher measures. And controls are things you keep the same. So you try to keep things the same so that they don't affect your results. So this may be things like the same age or the same organism type. You do these things so that they don't affect your results. In terms of graphing, your independent variable is going to go along the x-axis, which is this way, horizontally, And your dependent variable is going to go along the y-axis, which is up and down. So those are just kind of keeping reference as we move forward out of the E in sperm to the R. Before we finish through with the R, just two things to remember is that experimental procedures must be repeatable and they create evidence. So because they create evidence, they're going to give you data or results. If they don't give you data or results, it means you either have to make a new tool or you have to revise your procedure. So in terms of graphing and data analysis, we have something called raw data. So in the lab we just did, this would be kind of like your tallies. So this would be raw data. The data table would be now sorting this into something that everyone can access. So we could say 5 and 1. So the difference between raw data is it's kind of the tool that you're using as you're collecting the data. And the data table is now organized in a way that other people can access it. From a data table, you're then able to move to a graph. And when we think about graphs, for example, I just did dogs and cats, we think about something called tails. So tails stands for title, axes, interval, label, and scale. So what do these things all mean on a graph? You have a title at the top. Just like we said with the question, we want our title to be specific. It's not going to be something just called graph. It's a graph of what, in whose class, of what, in what time period, what year. Be specific in your title. Axes, that means more than just labeling X, Y. Tell me what's on that axis. Is it time? Is it category? Label out your x-axis. Same thing with your y. Is it number of penguins, number of turtles, number of cats, number of dogs? Be specific. Intervals is that they're equal spacing as you go up. So we want something that's 2, 4, 6, 8. Who do we appreciate? We don't want something that's 1, 52, 56. That's no good. We do not want that. We want nice, even intervals as we go up or to the side. In terms of labels, this might be something if you have a key. If you're doing a bar graph and you have a lot of stuff going on, labels kind of gives more information to the person looking at your graph. Lastly, we'll look at scale. It does me no good if your graph is this. And it's super teeny tiny and no one can read that. In the words of Ms. White, we need to use all the space. So fill your paper. In terms of scale, we want to make sure that our graph fills everything and is reasonable for the page. So just to summarize up, tails, title, axes, interval, label, scale. Be specific in your title, label your axes, have even intervals, use labels if it's confusing, and use all the space for your scale. Some additional analysis you might want to think about for probability. You might be doing things like mean, mode. If you take AP Bio, look at things like chi-square. If you take other classes and stats, there will be things like the t-test. There are other types of probability that you can use to analyze your data. You can look at other data and compare to it, and so much more. There's so many other data analysis tools, but right now we're just kind of starting with tails and maybe a little bit of probability. But a general trend is that the more data you have, the better your results. One data point is no good. We want to try to get as much data as we can. So we've gone through results, we've gone almost all the way through sperm. We just want to wrap up with one last one, making conclusions. So what does that stand for? RE stands for results with evidence and explanation. So you'll also get that handout with all the summary of the statements. But this is just some reminders. You're going to summarize what happened. PE stands for possible errors and how to fix them. So don't just say, I messed up. How would you fix it? Remember, there are a couple different types of errors. There are human errors, which you just fix them. There's nothing really you can do but do the project again. Do everything over. Human errors you want to try to minimize. There are tool errors, Or maybe you have a tool that only measures in metric and you need imperial units. And the last type of error is equipment, which is kind of related to tools or reagents, where maybe your enzyme is dead, maybe your plants are dead, so you can't measure them. But there's some possible types of errors. Lastly, we'll talk about practical applications. PA is the so what, who cares. Without practical applications, you're going to have problems. This is the money. Who cares? So what? Everything you do should have a basis and a motivation, a money market, something to drive it. So every lab should have something that you're thinking about. Why does it matter? Who would do this as their job? See it in an everyday application. That wraps up the scientific method and some of the things about graphing. So you should be able to analyze this drawing and kind of talk to me about some of these things that happened. In terms of our lecture, we talked about sperm and what sperm means. We broke sperm down. We talked about how sperm had to have smart questions. We talked about P, make predictions that are based on claims, evidence, and reasoning. For the experiment, it had to have an independent variable, a dependent variable, and a control. For the results, we talked about tails and different types of data. And lastly, for M, we talked about making conclusions using the REPPOW format. In conclusion, we find that the scientific method is repeatable, tested, and evidence-based. Using all these tools, we can make sure we document the process so that other people can get on the same path, find similar information to confirm our results. So hopefully you learned something about sperm tails and a little bit about the little parts in between the scientific method. I look forward to seeing you next time, and if you have any questions, please make sure to shoot me a message or talk to me on School Loop. Take care and have a great day.